So good morning. I'm Sue Ripers, Manager of Mission Support and Resources at Brain Tumor Foundation of Canada. As many of you are already aware, as I've had the opportunity to connect with most of you. Thank you for joining us today and taking time out of your weekend to be part of this event. Special thanks to the speakers, judges, teams, and attendees who have joined us. It's vital that we encourage talent and diversity of knowledge and research, and this event is a great start to a long relationship with all of you. Our research program and awards are designed to engage students, junior investigators, and established labs who are working collaboratively to defeat brain tumors and make a difference in the lives of those impacted. We're thrilled by the interest shown by undergrad students and look forward to seeing participants apply for future awards as they continue to advance in their education and careers. This year to date, we have awarded 2 studentships, 2 fellowships, 2 GBM feature grants, 1 elevation grant, and 6 education awards. And we currently have our $25,000 research grants opened, as well as our $2,000 Trevor Harrison Civic Engagement Scholarships. We too are very excited to support the Brain Tumor Surveillance Research Collaborative, formerly known as the Brain Tumor Registry. Its goal is to provide comprehensive data on the incidence, prevalence, mortality, and survival rates for brain tumor or for primary brain tumors, malignant and non-malignant. By collecting important characteristics such as tumor location, histology, sex, age, and region to better understand the patterns of occurrence and survival. We will not stop until every brain tumor is routinely counted, as every brain tumor counts. Our organization continues to support innovative collaborative research grants through the Brain Tumor Funders Collaborative, which drives progress in brain tumor research. Just this week, we awarded 2 liquid biopsy projects that were announced with a total of $1 million, showing that together we are stronger. This group brings together 4 core members: Brain Tumor Foundation of Canada, American Brain Tumor Association, the National Brain Tumor Society, and the Pediatric Brain Tumor Foundation. We are proud to be the only Canadian organization represented alongside this group of nonprofit, philanthropic, and advocacy organizations from the United States. We have also joined forces with varied organizations on the Decrypt grant, Defeating Embryonal Cancer in Young People Together. This research grant was a $600,000 grant awarded in 2023, and they've just recently begun the project after ensuring that some logistics and the involvement of multi-institutions were in place. This event, the Pam and Rolando Del Maestro Family Undergraduate Research Competition Awards, were established to promote scientific innovation among Canadian undergraduate students. I'm not sure that if our founders have joined us, but if they have, we welcome them, and we'd like to thank them for their continued support of our organization. The purpose of this event is to provide students with the opportunity to present their work as a team to today's diverse audience of students, scientists, researchers, healthcare professionals, patients, caregivers, to name a few, which focuses on a specific case related to brain tumor community. We had 16 letters of intent submitted this year, which were all incredible, and 6 teams selected to refine their presentation skills and receive valuable feedback from today's judges. We had one of our teams withdraw yesterday, so we do have one last team now presenting today. Today's teams will compete for monetary prizes, and from my experience hosting, there has been some institutional bragging rights as well. Thank you for being here. We welcome our judges, Drs. Megacy, Dr. Ranger, and Dr. Wang, who were instrumental with the development of the case and the review of the letters of intent and the selection of the teams who are here today, as well as those who will be asking questions and selecting our top 3 teams at the end of the competition. I would like to congratulate Dr. Wang, who took his final exam for the Royal College of Neurosurgery yesterday, and I'm sure that you are pleased to have that complete. And we're pleased you're able to join us today. So congratulations on that accomplishment, and we wish you best in the results moving forward. Each team is aware of the order that they'll be presenting today and has a team captain who will be responsible for ensuring you're presenting in the most ideal location, managing time, and sharing screens accordingly. For anyone who is the team captain, if you can please ensure that your name on your account is your name so that I know who to assign this screen sharing to. Each team is given 8 minutes to present, and you will then hear the following sound, which sounds like. Was everyone able to hear that?
Very faint.
Okay. Okay. Well, I will try and hold it up to the speaker a little bit better and ensure that I've got it on full blast. Once you
Can you
hear that
try it
sound,
again?
yep,
That was it?
yes, that's it.
Very, very faint.
I didn't.
Okay. So maybe I'll just indicate time is up.
That's much
Um,
clearer.
Yeah. And I think the teams are pretty good at managing their own time as well, but I'll apologize in advance for interrupting to say time is up when I hear a break and an opportunity that I can say so. So I will do that both for after the 8 minutes that the team has to present, as well as after the 5 minutes that the judges have, just to keep us on time. As judges are asking questions, we ask that the team who is up next that they start to preload their presentation so that that's set to start once the 5 minutes of questions is up from the judges. Thank you for everyone's understanding with the time frame and following the cutoff time to ensure that we do remain on time today. After all teams present, the judges will then deliberate. Judges will head off into a breakout room. And judges, I do not have a student this year to make sure that you get into that, but I know that if you have any concerns, you can reach out to me. And I will try and sort something out, or I trust that all of you are good to be able to sort something out if there's any sort of glitches in that, but should run smoothly. Um, if I can please get you to put your first and last name in the chat for attendance, and if you're sharing a system with someone, if you can put everyone who is with you, that way I can just ensure for the attendance tracking. Also ensure that if you're not a presenting team, that your mute is on, please. What else? I think we can jump right in. Judges, is there anything else before we jump into asking Dr. Ranger to review the case and the expectations of the team?
So, Sue, since
Hmm.
since the first team from Calgary are delayed, we're just going to go in order from team two on down.
We will. We'll start with McMaster, and then we'll end with the University of Calgary.
Okay.
Yeah. Okay, perfect. So Dr. Ranger, I will hand it over to you to share the case.
Okay. Um, Thank you, Sue. And thank you also to Dr. Wang and Dr. Megacy for sort of co-judging this along with me. So the case that was provided to formulate your research projects around is actually a patient of mine whose identity has been changed for the purpose of the competition. But this was a 7-year-old right-handed boy brought to our children's hospital by his father after having fallen with a sudden onset of right-sided weakness. And he was, in retrospect, fatigued, anorexic, and quite irritable over 2 or 3 months prior to that, with several episodes of headache. He had been brought to the local emergency several times and sent home. Sometimes his blood work appeared reassuring, but there was never any brain imaging performed, and he developed a clinical sign of intermittent strabismus involving his left eye and was referred to ophthalmology for evaluation, but had been brought into the emergency department before he had a chance to be seen by his eye care specialist. So on the morning of admission to hospital, he fell after having dragged his right leg and was unable to brace himself, became unbalanced, and he struck his face when he fell down. At that point, a CT scan was done, and you've all had pictures of the images from that scan showing an abnormality in the child's left hemisphere, and that was followed by an MRI scan that better defined that abnormality. And it revealed a complex solid and cystic lesion involving quite a large expanse of the child's left hemisphere, but mainly centered in the parietal and occipital lobes, with extension across the midline through the splenium of the corpus callosum and midline shift. It enhanced with gadolinium, and there was necrosis and hemorrhage associated with it. And he was referred for pediatric neurosurgical consultation urgently. The team met with the child's family, discussed the potential radiographic diagnoses, and options for management were discussed. And the child's family were provided with the Brain Tumor Foundation of Canada's Handbook on Pediatric Brain Tumors, which is an excellent resource for families, usually offered up front right at the time of diagnosis. Parents agreed for urgent surgery and attempted resection of the brain lesion to acquire tumor samples for diagnosis and potential for debulking, but not necessarily full resection. So he underwent the following day a craniotomy for partial resection of the brain lesion. A frozen section or quick section was done that confirmed a lesional sample consistent with a glioma, and further detailed pathological review returned the diagnosis, which is your research problem, of pediatric type diffuse hemispheric glioma, H3G34 mutant, and WHO grade 4. So there were a number of potential questions posed as potential research projects, and we had a number of submissions of projects, and your teams have been selected as the top teams with research ideas to address a problem like this.
Thank you, Dr. Ranger. So we will have the first team, McMaster 2, Veer and Vanny, if you can load your presentation. Excellent, thank you. And whenever you're set, you can begin.
Okay. Hello, everyone. My name is Vani Chaudhary, and I'm here with my partner, Veer Chaudhary. Our proposal focuses on dual-pronged immunotherapy and experimental intervention for H3G34 mutant pediatric type diffuse hemispheric glioma, or PDHGG. So, PDHGG is a rare and aggressive brain tumor with a poor prognosis and limited treatment options. Notably, H3G34 mutant tumor cells exhibit epigenetic changes at pro-inflammatory gene loci, and this creates a relatively immune-permissive tumor microenvironment, which suggests that these tumor cells might be extremely sensitive or more sensitive to immunotherapies.
An immunotherapy that has been proven to be effective for solid-state tumors, becoming the first FDA-approved cellular treatment for such cancers, is tumor-infiltrating lymphocyte therapy. It functions by extracting lymphocytes in the tumor microenvironment, expanding them, and re-administering them. Remarkably, TILs innately have a diverse range of tumor-specific antigens, which is a roadblock in other adoptive cell therapies. They can also be engineered to target a broader spectrum of neoantigens, which are tumor-specific mutation-derived proteins, to improve tumor recognition. However, its usage in PDHGG is limited by the cold tumor microenvironment, which causes T cell exhaustion, a state in which T cells which attack the tumor become progressively dysfunctional. And the cold TME is recognized by a high representation of immune checkpoint inhibitors and interferon signaling, which both cause T cell exhaustion.
Now, we're interested mainly in three immune checkpoint inhibitors, LAG3, CTLA4, and PD-1. So essentially, these immune checkpoint inhibitors work by binding to an activated T cell and sending an inhibitory signal, which leads to T cell exhaustion and a decrease in cell proliferation, which decreases the immune response against the tumor.
A transcription factor of note is IRF2. IRF2 is the primary transcription factor responsible for interferon-mediated T cell exhaustion. And overexpression of IRF2 promoted tumor cell migration and invasion. And the deletion of IRF2 actually improved responsiveness to adopted cell therapies and limited the invasion of gliomas.
One very promising approach that has emerged is ADTK immunostimulatory gene therapy. A study by Fabiani et al. Reported 50% long-term survival in mouse models and an increase in anti-tumor immunity and immune memory. It is thought that this therapy drives a shift from a cold tumor microenvironment to a hot tumor microenvironment, which therefore enhances T cell activation and reduces T cell exhaustion. Now, this brings us to our research problem. TILs have shown potency in several solid-state tumors. However, comparative efficacy has not yet been achieved in PDHGG. So to address this gap, we propose the usage of neoantigen primed TILs with a combinatorial knockout of the immune checkpoint inhibitors PD-1, CTLA4, and LAG3, as well as the transcription factor TRF2, and so we co-administered with ADTK immunostimulatory gene therapy. We hypothesize that co-administrating these enhanced TILs with ADTK gene therapy will synergistically overcome the immunosuppressive tumor microenvironment to significantly improve clinical outcomes in PDHGG patients. So moving on to our specific aims, we have three. The first is evaluate the efficacy and safety of unmodified and modified and enhanced TILs, determine whether enhanced TILs independently and in combination with ADTK gene therapy reduce cell exhaustion and enhance tumor infiltration. And number three, assess the long-term therapeutic efficacy, synergy, and safety of unmodified TILs and enhanced TILs co-administered with ADTK gene therapy. So moving on to our methodology. So we plan on using 125 mice from birth to death, and we are going to split these into 6 cohorts, each receiving a different type of therapy, as seen in the diagram. Now, moving on to phase 1. So we need to create a tox expression reporter model because tox is a transcription factor associated with T cell exhaustion. So to create this, we're going to take single cell embryos and we're going to inject them with a CRISPR-Cas9 complex and nanoluke cassette. And we're then going to implant this into a pseudo-pregnant female. And then once they give birth, the pups are going to be confirmed to be nanoluke positive. And once they are, we're going to introduce the H3G34 mutation using the Sleeping Beauty transposase system.
Moving on to phase 2. First, we'll collect peripheral blood samples, which will undergo centrifugation, after which the lymphocytes will be cultured in media, and media-adherent cells will be expanded with interleukin-4 and colony-stimulating factor, which will generate immature dendritic cells. After that, one tumor sample will be obtained and cleaned, and the sample will be minced, run through a cell strainer, and undergo centrifugation, after which the tumor-invading lymphocytes will be isolated and transduced with the lentiviral vector encoding Virulina luciferase so that we can visualize and measure the TIL infiltration. After this, it'll be cultured for 72 hours to allow for protein expression, and that will be described later on. For cohorts 3 and 6 specifically, a second tumor sample will be required, and the sample will be minced and lysed via freeze-thaw cycles, after which larger particles will be removed with centrifugation. After that, it'll be filter sterilized and split into aliquots. And the lysate will be added to immature dendritic cells so that the dendritic cells can manifest the specific neoantigens of the tumor. After that, it'll be tumor necrotic factor alpha will be induced maturation, and the dendritic cells will be quality checked via flow cytometry. Moving on to phase 3, the TILs and the dendritic cells will be co-cultured and incubated so that the TILs will be primed to those specific neoantigens. And the neoantigens will be verified via flow cytometry, after which they'll be electroporated, and CRISPR-Cas9 will be used for the knockout of PDCD1, CTLA4, LAG3, and IRF2, after which both the enhanced TILs and the regular TILs will be cultured in a solution of interleukin 2, 15, and 21. Moving on to phase 4, All therapies and therapy combinations will be delivered into the tumor site via convection-enhanced delivery, directly bypassing the blood-brain barrier, which serves as a significant roadblock with the delivery mechanism of many cellular therapies and pharmaceuticals.
Now moving into phase 5, data analysis and collection. So we'll be tracking the overall and median survival for each cohort and then constructing Kaplan-Meier curves. We'll also be tracking tumor burden, TIL exhaustion, and TIL infiltration, and this will be observed via bioluminescence imaging. So we'll be taking baseline images before going into phase 4 so we can observe the changes in the tumor environment. The baseline image will then be followed by weekly scans to monitor tumor burden and tox expression and TIL infiltration. This data will then be analyzed via a two-way ANOVA, two-case post-hoc test, and Pearson's coefficient. Furthermore, every week after week 4, multiplex speed-based assays will quantify cytokine levels in peripheral blood samples linked to CRS and cytotoxicity with two-way ANOVA for data analysis. But why does any of this matter? What's the clinical relevance? Well, the study assesses the unmet need for efficacious treatment in PDHGG. This dual therapy aims to increase tumor regression, reduce T-cell exhaustion, and extend survival. Therapeutically, this project offers a novel strategy that could significantly improve clinical outcomes for patients suffering from these gliomas, like Jacob. Successful demonstration of safety and efficacy in this model will lay the groundwork for translating this combinatorial immunotherapy into clinical trials, advancing precision immunotherapy in brain tumors, and potentially redefining the standard of care in H3G34 mutant pediatric type diffuse gliomas. These are our references. We'd now be happy to answer any questions that you have.
And that was right on the buzzer. You guys were good for timing. Judges, I'll open it up to you.
Hi, it's Adriana here. So thank you, Veer and Vanny, for submitting this project. Very interesting and complex with a number of different layers to it. I think I understood it best trying to go through your letter of intent, the written part of your project, and it's nice to see the visuals of your presentation very clearly presented like that. So as I understand, you're trying to use two immune strategies. So you're trying to use tumor-infiltrating lymphocytes to boost a child's own tumor-fighting T cells, and then you're using the 80 TK FLT gene therapy to heat the tumor up so the immune cells can work better. Is
Yeah,
that
exactly.
Yeah, that's exactly what we're doing.
okay?
Yeah.
That's sort of what I got out of the letter of intent mainly, and then I appreciate your methodology much better. So it raises a few questions. One of them is really sort of a safety risk consideration for this type of approach to tumor therapy, because immunotherapy has risks like cytokine release syndrome and off-target effects, or even neurotoxicity. So how would your study design maybe account for or measure adverse effects? And how might that influence clinical applicability?
Yeah. So we do take peripheral blood samples every week and run cytokine assays on them to measure the peripheral blood and see if there's any signs of like cytokine release syndrome. Notably, though, the reason that we chose to go with TILs was because so far TILs haven't shown any significant cytotoxic effects, like, for example, the CAR T cells have. And so it has generally proven to be a lot more safer, I'd say, and more effective in terms of attacking solid state tumors.
Yeah, and just sort of adding on to that, sort of the neoantigen phase, where we kind of let the dendritic cells present this to our TILs. It's supposed to improve their recognition of tumor cells specifically, and so hopefully this will also limit sort of the off-target effects that we'll experience.
Yeah, and another key difference is it's also the way the TILs work. It's not just one receptor that we're looking for, because TILs generally they already have a diverse range of multiple receptors that they use to recognize the specific tumor, and so yeah, so it makes it a lot more precise in terms of targeting the tumor and limiting the off effects.
Okay, thank you for that. And I guess the other question that I had, if I'm allowed to ask another one, is just, you know, you have some endpoints that you're looking for, and I just wondered what sort of clinical endpoints might be meaningful to demonstrate benefit, you know, from your sort of preclinical measures like tumor regression, TIL infiltration, and that sort of thing.
Do you want to take this one or should I take it?
Yeah, so basically what we'd be looking for is deeper TIL infiltration and a reduction of tox expression. And so basically what that just say is that the TILs would be able to spread more evenly across the tumor and that they would actually be more effective in comparison to our control if that's what we see. And so what that would mean is that especially for these kinds of tumors where they have a really cold tumor environment, this would allow them to really be effective in those cases and reducing tumor burden. I'm not sure if that fully answered your question, but
it does. It sort of does. I was kind of looking for something might be more like clinical, like, you know, what would it mean for a child down the road? But I think
yeah,
that's
okay. In
fine.
terms of
Anyway, thank you.
longevity of the response as well, the TILs also have a very lengthy response. So if they do have the tumor and it does come back, the TILs are already primed for that specifically. And so what it means is that they'll be really good at ensuring that the tumor stays gone, essentially.
Okay, so I'd actually like to take this question. So essentially, the idea is that this specific gene therapy is really good at ensuring that your immune system recalls the specific sort of mutations in the tumor and the antigens that it produces. And what they found is that there was like a reduction in the number of relapse rates. So when you have such a sort of refractory tumor that is very prone to relapse, such as PHGG, this sort of immune response should hopefully prevent relapse, and that should be indicated hopefully through our through what's called right here the reduced tumor burden and also how spread out they are within the environment.
So related to that then, so do you anticipate that this will be a treatment that you have to give for a prolonged period of time, or that you would have to give it indefinitely? And would you anticipate that the neuroimaging would improve or would just stabilize?
So I actually did look into this. So for this sort of immunotherapy, the minimum amount of time it takes to work is 3 weeks. And we think that in addition to the sort of genetic therapy that we're giving, that could potentially expedite the process. And given that we're already part of the process is taking a tumor biopsy, we would recommend doing this in combination with some sort of surgical resection. And to answer the second part of your question about what we hope to see in terms of neurological imaging, we're hoping to see like a complete reduction and it should just stabilize at that. And if there are any sort of relapses, it should be able to manage that with the sort of immune memory that's been created.
Yeah, and it'd be one dose. It'd be one administration, and that should provide the immune protection that they need.
And what potential side effects or adverse effects?
So as far as we could find, like in terms of the TIL therapy, there actually haven't been many recorded cytokine release syndrome or any of those other effects. So the main side effects that are associated with TILs are mainly just due to the radiation that you get beforehand. And which makes it so what we expect to see is very limited side effects, if any, and at a very small dose.
Ryan, do you have any questions?
How much time do I have?
We're actually out of time.
Oh, okay.
Can't you give Ryan more questions? Waiting.
Yeah, no, go ahead.
Oh, okay. Sorry. Yeah. No, great presentation. Very well structured and ambitious too. So I just wanted to clarify, how are you building your tumor model? Are you inducing the tumor in those mice, or are you
Yeah.
using patient samples?
So we're actually inducing the tumor in the mice. I actually took this from another study, which was studying this like PDHGG gliomas, and they uh, induced the tumor in the exact same way.
Okay.
And I am also inducing specifically H3 G34R, which is like the primary mutation in I think it was 90% of PDHGG cases.
Okay. And what challenges do you foresee doing that in a mouse model versus, say, an actual patient-derived tumor sample?
It's different because biologically they're different organisms. Again, that I feel like that would be a uh, potential topic of study in for further study
Yeah.
after the sort of initial
Yeah,
um,
but we do see that like it could be applied both ways as well. I think if we were to use a human sample as well, we think that that would just be the little slight change we would make in our methodology would still stay roughly the same. So yeah, and it just in this case we decided to do it just for ease of imaging and in terms of the methodology.
Great, thank you.
Excellent. Thank you very much. Queen's University is up next. What I will do, since I don't want to interrupt people, is just to put up a white screen when you have 1 minute that's remaining on both the presentation and the questions. And when you see the white paper again, that will mean that time is up. So I will leave it to Queen's University 1 team to start presenting whenever you're set.
Perfect. You guys can see my screen, right?
Yes, we can. Thank you.
Perfect. All right. Yeah, Mohammed, can take it.
Hey everyone, today we will be presenting our research proposal which covers the topic of targeting transcriptional condensates using SE gene computational analysis in H3G34 gliomas.
So to briefly summarize the case, we have a 7-year-old who was diagnosed with an aggressive H3G34 glioma after sudden collapse. Even with current treatment, median survival is typically about 18 months. And so our proposal aims to develop a targeted therapy that could extend survival for children like Jacob.
So let's first understand this tumor. Biologically, the G34 histone mutation in the tumor acts in cis to impair H3K36 trimethylation, which disinhibits PRC2, a gene silencing complex, raising H3K27 trimethylation along with rewiring lineage programs. And that's what you see happening in Figure 1. These tumors frequently carry PDGFRA gene amplifications, which allow for heightened cell growth, as well as mutations in TP53 and ATRX tumor suppressors, and adopt interneuron-like transcriptional states. Together, this creates reliance on super enhancers, which are DNA regions packed with transcription factors along with coactivators that drive high oncogene output.
So moving on to the rationale, those super enhancers that Ahmed mentioned on the last slide, you can actually visualize it here. So in the diagram, you can see how these super enhancer regions and all the transcriptional factors that it recruits. And so key players we want to mention here are BRD4, a protein which binds acetylated histones to recruit transcriptional machinery, and mediator 1, which links enhancers to RNA polymerase 2. These factors then come together and cluster into droplet-like condensates inside of the nucleus, and they act almost like RNA factories. And so what we believe is that disrupting these condensates by blocking factors like BRD4 can silence many of those cancer-driving genes. For example, previously in H3K27M diffuse midline gliomas, BET inhibitors shut down transcription and halted growth. And so we believe that H3G34 tumors share this same vulnerability, and we can act on this target.
So just to recap, we're hypothesizing that H3G34 mutant gliomas are critically dependent on these super enhancer condensates, and disrupting these condensates will impair tumor growth. Our objectives are threefold. Firstly, using SEG and AI-augmented tool to map super enhancers in H3G34 gliomas. Secondly, to disrupt the transcriptional condensates in vitro. And then finally, to evaluate therapeutic efficacy in vivo.
So our first aim is to actually identify these superenhancers that drive tumor growth in H3G34 gliomas, and then confirm whether they form transcriptional condensates. We begin by looking for active enhancers in the tumor genome. H3K27 acetylated regions signal whether DNA is active, and to find these sites, we will use chip sequencing. This is a technique that involves cross-linking DNA to proteins, shearing it into fragments, and using an antibody against H3K27 acetylation to pull down the acetylated pieces. This gives us a map of active enhancers in the tumor cells. On the other hand, we also need to know which genes are actually being expressed. For this, we use RNA sequencing to measure all of the mRNA molecules in the cell, and this indicates which genes are turned on and how strongly they're expressed. So in simple terms, the chip sequencing tells us where the enhancers are active, and the RNA sequencing tells us which genes are being transcribed. Now, the thing is, we don't just want to find those enhancer regions. We want to find super enhancer regions which are functionally important. And so this is where the next step comes in. A newly developed AI model by Shinkai et al. Called SE-Gene, short for super enhancer gene, integrates that chip sequence enhancer data with the RNA sequencing expression data. Using these data sets, it asks the simple question of, does this signal at a enhancer rise and fall with the expression of nearby genes across samples? If the enhancer activity correlates with gene expression, SE-Gene keeps it. If not, it's filtered out. And so this is what gives us our candidate super enhancer list in HCG34 gliomas with direct gene associations. SE-Gene builds a super enhancer to gene network and highlights those key hubs. In our last step, we test whether those super enhancers actually form transcriptional condensates. And in gliomas, this is done with immunofluorescence microscopy. If BRD4 and MED1 appear as bright foci in the nucleus at predicted super enhancer sites, this indicates condensate formation, which will act as our target in AIM2.
AIM2 entails disrupting the identified super-enhancer regions which form condensates, and this process will be twofold. The pharmacological suppression of super-enhancer-driven transcription in vitro, along with CRISPR-Cas9 interference to epigenetically silence selected super-enhancers. Pharmacologically, we will treat the mapped condensates with the BRD4 inhibitor JQ1 and the CDK7 inhibitor THC1. We expect JQ1 will kick BRD4 off the super enhancers, as Zhang et al. Showed that BET inhibitors cause BRD4 to disperse from nuclear foci. The CDK7 inhibitor, on the other hand, works more globally by halting transcription initiation. Moreover, prior work shows THZ1 kills glioblastoma cell lines, along with broad transcriptome suppression and downregulation of receptor tyrosine kinases, including PDGFRA. For instance, as shown by the dose response in Figure 2, THC1 demonstrates sub-100 nanomolar potency and downregulates PDGFRA mRNA as well as alpha protein expression as indicated by Figures 3C and 3F, thus potentially impairing tumor growth. Finally, we will deploy CRISPR-Cas9 with guide RNAs targeting the enhancer regions of top candidate oncogenes and track outcomes to see if this will repress enhancer activity and mimic condensate disruption by genetic means. If successful, this option can move to animal trials.
In AIM3, we'll test the lead candidates from AIM2 and whether our strategy works in vivo. We'll create an orthotopic xenograft model by implanting patient-derived H3G34 glioma cells into the brains of immunodeficient mice. We'll deliver treatment using the lead inhibitor from AIM2, which could be JQ1 or THC1 or a combination. To test efficacy, we'll track tumor growth with MRI, and what we should see is that treated mice should live longer and have smaller or slower growing tumors. We'll also test safety, which is critical for pediatric use. Mice will be monitored for weight loss, behavioral neurological changes, and organs examined for toxicity. If survival improves with acceptable toxicity, we'll be ready to pursue a phase 1 trial.
So in conclusion, this treatment option is significant because the H3G34 mutant remains lethal with limited options, and disrupting super enhancer hubs presents potentially a rational vulnerability. Furthermore, AI augmented tool mapping of super enhancers enables patient stratification along with personalized treatment with tools such as CRISPR-Cas9 for patients such as Jacob. Complementarily to the mapping, we have shown a path with transcriptional CDK7 and BET inhibition that could potentially halt tumor growth via enhancer suppression and condensate disruption. Going forward, our goal is to carry super enhancer targeting into the brain directly and make it clinically actionable for H3G34. Figure 4 maps how drugs cross the blood-brain barrier and why many fail to do so. In the case of JQ1 and THC1, this should not be a problem. However, if another inhibitor is more well suited to disrupt the condensate and cannot traverse this barrier, then measures must be taken to permit this, such as maybe using focused ultrasound or attaching permeable nanoparticles such as liposomes. Thank you very much for listening to our presentation. Here are our references, and we'd be happy to take any questions.
Excellent. Thank you very much. Judges, I'll open the floor to you.
Well, I started last time. Maybe somebody wants to go ahead of me this time.
Sure. So as you're proposing it now, how do you plan to administer the therapy in a human patient?
So currently we haven't actually looked into the administration in a human patient. So normally these BET inhibitors, they typically come in oral form or a injectable. But when our plan is for these mice models is to deliver them intraperitoneally, or we can use a combination of oral dosing. And then as it goes through those clinical trials and we see kind of the most safe and it's a safe way of administering it, whether that's IV, whether that's oral. We'd have to kind of find that correct dose and go through that whole process of finding the toxic dose and the acceptable dose that's applicable for patients, and seeing how that works out. But yeah, for now we haven't figured that out yet.
I mean, your last sort of slide there showed the blood-brain barrier issues with that. I mean, you were talking about possibly direct injection or somehow nanoparticle delivery. Is that what you were getting at there?
Yeah, so I know I think with the case with THC1, for example, that is able to cross the barrier, but we're not sure if like all the inhibitors, for instance, are able to do so. And so we're just saying that because those are like overall tool compounds, we want to limit offset targeting as well as potential adverse effects and toxicity as much as possible. And so more direct approaches could potentially be something such as like wrapping it in like nanoparticles that could cross, as well as using um, um, the other technique I said, fluorescence,
Ultrasound?
which, yeah, ultrasound, sorry. Yeah, it's to sort of create those small holes in the brain and then allow it to pass directly.
There's another way that you might do it. Can you think of that?
Um, I, I,
Three
I
judges,
actually,
three judges might be able to help you.
I,
Might be able to help you.
I think I actually read something about it. It was like convection something, if I'm not
Yeah,
mistaken.
convection enhanced delivery,
Yeah,
or
convection has over, yes.
there can be uh, sort of membranes laid down at the time of surgery that can be slow release. They've had trials in the past. They've had some problems, obviously,
Yeah.
but there are other ways that surgeons can help you get drugs to a tumor cavity after resection. Ryan, Adriana?
Yeah, I can go. No, thank you very much for your presentation. I think for my question, I just want to go to your methodology really quickly for AIM-1. Um, so I'm not sure if you mentioned this, but what samples are you using for, I guess, your step 1 and 2 of AIM-1? You're doing your RNA-seq and chip. How are you, what cells are you using for that?
Pretty much what we're using in step 1, we're using patient-derived G34 glioma samples. So this could be either the primary tissue itself, or take those patient-derived cell lines. And by doing this and running that chip sequencing to map those enhanced regions, or by using the RNA sequencing, we can see exactly where those super enhancer regions are, and if they are mapped to a gene, and if those genes actually turn on. And so pretty much what SE gene does is, so before there was a technique called ROSE, which is ranked ordering of super enhancers. So these only check the signal strength, but they don't check if it kind of regulates the genes. And so what SC gene does is it combines those two factors and finds exactly if those super enhancers connect to a gene. And so pretty much we take those patient-enhanced G34 glioma samples, run it through this process, and we're able to find a network graph. So yeah, that's kind of the source of those samples and how we're using them.
Awesome. Thank you.
Mhm.
Let's see. Hello, it's Adriana here. Thank you for sending us this project and then describing it very nicely. These super enhancer inhibitors, BET and CDK7 blockers, can affect normal cells too. So how do you distinguish tumor-specific vulnerability from kind of global transcriptional suppression? Like what strategies could you use to minimize toxicity from this?
Mohammed, I think if you could elaborate a little bit on this.
Yeah, so these inhibitors can absolutely hit transcription in normal cells too. The difference is that tumor cells with huge super enhancers are well, they're basically far more dependent on that constant transcriptional drive. So it'd be kind of like cutting the power. Normal cells can switch to backup circuits, but the tumor is wired to like essentially like one massive breaker and collapses if you flip it off. So to make this distinction experimentally, we could include, I guess, like normal astrocytes or like wild type glioma cell lines as controls, and then compare the transcriptional readouts. What we're hoping to see is a much narrower like safety window in the tumor lines.
Yeah, and just connecting on this actually goes back to the first question, which is asked by Joe there. We're going to try and target that delivery on those tumor cells by using those nanoparticle formations so that we can concentrate the drugs and minimize the systemic, the overall exposure. And along with this, we're taking a dual approach. We're not just taking a pharmacological approach. We're also testing, by finding those super enhancer regions, if CRISPR-Cas9 can cut out those regions and pretty much disrupt those condensates. So we're taking a dual approach, which is supposed to be targeted. And that's actually the kind of main aim of our project, if that answers the question.
Oh, that's good. Thank you very much.
Mhm. Thank you so much.
Thank you.
Thank you very much. I think that we have University of Calgary that's all set. So whenever you're ready, please get started.
Just waiting for my partner, yeah.
Hello, everyone. I'm here to present our solution to one of pediatric neurosurgery's most pressing challenges, which is complete resection of H3G34 mutant gliomas while preserving neurological function. Our innovative idea is to use AI-guided Raman spectroscopy to provide neurosurgeons with a molecular vision during operation, while also answering one of Jacob's parents' most critical questions, which is, are there any surgical adjuncts at the moment that will allow total resection of glioma? I want to first draw your attention to the stark reality. Children like Jacob, who's only a 7-year-old, are faced with a median survival of 12 to 18 months only. H3G34 mutation is found in about 15% of high-grade pediatric glioma, which spreads like roots in a healthy brain. It has an infiltrative nature, so it's hard to distinguish healthy brain cells from the infiltrative tumor cells. With the current imaging technologies, it is nearly impossible to reliably, completely distinguish healthy cells from tumor cells, which means that surgeons are essentially operating blindly. So just think about how critical it is to find a guide that is going to help us completely resect the tumorous cells from the healthy cells, because for a developing child, every millimeter matters. We propose developing an AI-guided Raman spectroscopy that acts as a molecular vision for the neurosurgeons. How it works is H3G34 mutation creates differences in the molecular structure of the brain. When we shine laser to any molecular structure, it creates Raman peaks, which are specific to specific molecular structures. What we plan to do is use Raman spectroscopy to create Raman peaks, which are specific to H3G34 glioma mutant cells. And then we're going to train our AI system to interpret those signals right away so that it gives our surgeons a color-coded map showing tumor versus healthy cells in real time. This goes beyond any current imaging capabilities. Our hypothesis is our system will achieve over 95% accuracy in distinguishing tumorous cells from normal brain tissues, which is going to eventually increase the survival outcomes of children like Jacob. Our first research aim is to develop an H3G34 specific spectral library. Essentially, what we will do is we're going to shine the laser on tumor core, tumor margins, as well as healthy brain cells, which is going to create an array of spectral data, which we're going to organize in order to identify at least 5 to 10 distinctive spectral peaks using principal component analysis. Then we're going to use that data to train our AI to distinguish tumor cells from healthy cells in real time. We're also going to validate the efficiency of our system using gold standard histopathology.
Our second aim transforms spectral data into practical surgical tool with two key innovations. First of all, we're implementing a transformer neural network, as shown. Second, I think I skipped a slide. First of all, we're building a transformer neural network architecture, basically leading edge of today, which outperforms the CNNs, which I mentioned in the LOI, for special pattern recognition. Second, this novel approach we're developing, phased array Raman spectroscopy system. So we basically focus the beam, so like you'd have the probe, and we change the focus of the beam. We'd have like a fiber optic array of the transducers, and it would change the focus and scan the tissue like as you're moving it across the brain tissue in real time. It'll pass it by the AI system. And the hope is it would integrate successfully with current navigational platforms and achieve real-time analysis. And you'll have like a color-coded system where you'd have like green for like good tissue, yellow for like doubtful tissue, and red for like probably healthy brain tissue. And we can develop the margins for what's healthy and what's not healthy based off histopathology and a lot of experiments. Our third aim validates this technology in living systems using the avatar model approach. We'll establish orthotopic xenografts using patient H3G34 mutant cell lines, as shown in this diagram. This is from a paper. We can these models, like they're developed by Drew and colleagues. They faithfully recreate the infiltrative growth we expect. We can split them into two surgical groups. Conventional white light visualization versus Raman guided dissection. We'll measure residual tumor volume using post-operative MRI and tac survival curves based on similar studies. We expect a 40 to 60% reduction in residual tumor and a 25 to 30% improvement in median survival. Let me walk you through our methodology and validation approach. For machine learning, we'll use an 80-20 training validation split with 5-fold cross-validation. The ROC curve basically distinguishes between like how many false positives are there versus how many positive falses are there. And the transformer architecture shown here processes special data through multiple attention layers, identifying subtle invisible patterns to conventional analysis. It will use GPU acceleration live during surgeries to immediately process the data that comes out of the phased array Raman spectroscopy. For preclinical validation, orthotopic xenografts will undergo mock surgeries and will compare outcomes versus between conventional and Raman-guided approaches, measuring both residual tumor and survival endpoints. Our expected outcomes directly address Jacob's parents' questions about surgical adjuncts. First, molecular signatures will identify 5 to 10 distinct Raman peaks, differentiating H3G34 mutant tissue from normal brain with greater than 95% accuracy. Second, our surgical integration, 3D or 3D color depth maps shown here integrated with cranial navigation systems will provide real-time tumor visualization. Third, our clinical impact, which will be 40 to 60% reduction in residual tumor, translating 25-30% increase in median survival, potentially giving children like Jacob an additional 3 to 6 months or even more. Critically, we expect an 85% reduction in post-operative neurological deficits by preserving healthy tissue. Brain Tumor Foundation of Canada. Today, I present to you a novel approach. There are so many 300 to 400 children annually diagnosed in North America. Even modest improvements in surgical precision could save dozens of lives yearly. Our technology transforms impossible challenge, seeing invisible tumor cells into solvable problems, especially with the phased array system, which scans the tissue dynamically as you're going. These are our references, and you can ask us questions.
Thank you. Judges, I will pass it on to you.
So go first, I guess, or
go ahead,
Ryan.
Yep,
okay, yeah, sure. No, thank you for your presentation. And, coming from the neurosurgery side, this is, something that's been in the works for a little while now for various different tumors. So, thank you for bringing this up. So first question, can you go to your methodology really quickly? I just want to see AIM-1, I guess. I think you're looking at how many, determining how many samples you do and sort of the building your library. So do you think 50 samples is sufficient for you to build a library that would give you the accuracy that you would need? And are you going to use other tumors, say like an adult GBM or a medulloblastoma, to then see if your system can accurately, differentiate between those types of tumors?
So the goal here, or right now, it's not to distinguish between different kinds of tumors. It's to distinguish between healthy tissue and like how H3G34 tumor behaves. So if you look at the other tumors, they might exhibit similar characteristics in terms of how they behave. So they might be detected by the system. And 50 patient sample, it's kind of quality, not quantity of samples. Like modern AI systems are basically trained on the whole internet, but they're nowhere close to a specialist level of knowledge or expertise. So it's kind of quality of the data. And of course, we can scale our data as we go up, and it's about the quality of the features that we can extract. Well, the neural network approach we're going to use, basically, it's like nodes and edges. and we open up the space into a much higher dimension, which helps us extract features that are basically not visually distinguishable either to the naked eye or using even normal AI systems. So this is like a very high-end approach.
Okay. And then just another quick question for you. So, Raman spectroscopy has been used for other tumors, and I don't know how much reading you've done into the topic. There's things like fast glioma now that's been introduced, and various other techniques. What makes your approach different or potentially more effective than other Raman spectroscopy
So
techniques that have
I've
been
read
described?
about a bunch of systems that are currently in use or like being experimented. What I am proposing is a phased array system. So instead of the normal approach where it just scans a single, like the surface or the tip of the tissue, this approach will scan like into the depth of the tissue using like it'll use multiple transducers, and like it's gonna scan. So let's say you have brain tissue right here and you have the scanner. You put it up against there, and it's gonna dynamically change the focal point using physics and a bunch of complicated math to basically scan different points of the tissue. So not only do you have like information at the surface level, you have depth information, which has never been done before.
Okay, thank you very much.
So
Any other questions?
I'll just follow on Ryan's train of thought. So in Canada, McGill has done some of the work on Raman spectroscopy and presented some of it. So in your concept of this, how would the surgeon use this tool? Give us an example. So we're operating on somebody's tumor, and we have this device. How would we be using it? How would that guide what we're doing in your mind right now?
Okay, so you already resected most of the tumor that you've seen on the MRI, and you're at the barrier of tumor to healthy tissue. You grab this device, and you just probe it around, and it'll like it works in conjunction. It will hopefully work in conjunction with the systems, the navigation systems that you have in the operating room. So you would basically run it across the tissue up and down, and it would create a map of the whole area, and basically it would predict tumor tissue versus healthy tissue, and like the margin with like a certain confidence at certain intervals and distances, and you'd have like a map of how deep to resect and like where to resect exactly. That's the aim.
And I mean, could we put this device on the tumor right when we start, or would there be too much tumor for it to? Would it be overwhelmed by the tumor, or would we have to use our own judgment as to when we think we've reached the?
It would detect the tumor cells. It is designed to detect. So basically, if you put it against a tumor, it sees all tumor. It should basically give back on the monitors full-on tumor. That's the idea.
And this is occurring in real time. It would show us
the aim is for it to occur in real time.
Okay.
Currently, maybe you'd be looking at like half a second to a second, like a delay between actually mapping it to seeing it on the screen. But even with that, it should be very operable, and reducing time on the table will improve patient outcomes. And yeah.
Thank you.
Yeah.
Hi, it's Adriana. Thank you, Ikra Omar, for bringing this idea forward. And you have, just by chance, a bunch of surgeons on your judging panel here. So, and I found your second slide quite humorous. I think we've all had that feeling that your surgeon in the picture is kind of expressing that kind of wide-eyed, now what do I do? So you propose greater than 95% accuracy, actually, in distinguishing these glioma cells from normal brain tissue. But my question is really sort of along the lines of technical feasibility or like the translational hurdles of that, because the intraoperative environment can be really, contaminated with noise potentially, like blood or like the artifact of cautery, especially these tumors are quite bloody. The tumor tissue is heterogeneous. So how do you ensure that your Raman system maintains its accuracy under real surgical conditions?
So, well, there's already a paper actually about this, and they're using, as you guys are also pioneering in the Raman spectroscopy. If I can send you the paper. It's basically they achieved 92% accuracy with 86% specificity for the tumor, uh, which is with Raman spectroscopy. I'm not sure if that was interoperatively, but I read a couple papers about interoperative Raman spectroscopy, and they've also achieved a very high level of success with it. So the phased array system will just like increase your accuracy and the depth of data that you can see, and on top of that, it is trained on multiple types of data. Like so the way we acquire data is we train it on an operative environment. We wouldn't train it on just random tissue. Like data would be collected. It's like as you're taking tumors, we'd also take Raman spectra, and this data would be gathered over time and then compiled into a functioning system, hopefully.
Okay. And then how do you think something like this kind of intraoperative tool, I guess, would compare to stuff that's already in use, like 5-ALA?
Oh, is that a Raman spectroscopy probe?
Well, 5-ALA fluorescence is trying to show us residual tumor in a resection cavity. I mean, do you think this has an advantage over something like that?
It would just fluoresce, and the tumor does it work based off an injectable dye that only shows you the tumor cells, or how does that?
It's an oral agent the patient takes within a time frame right before surgery.
Mhm. Well, in theory, this would probably have better outcomes when it comes to tissue classification, much better than fluorescence. Maybe I'm not sure how well this specific drug infiltrates like the blood-brain barrier and then goes into the tissues, and then it would have to fluoresce. I have not actually read any papers about it, but it is interesting. But realistically speaking, even with this fluorescent approach, you know where to cut, but you don't know how deep to cut, is my point. Because
Okay.
millimeters can mean the difference between neurological function and non-neurological function.
Yes, that's certainly true. I think, you really risk function loss as you try to gain extent of resection sometimes.
Exactly.
Yeah, thank you.
No problem.
Excellent. Thank you very much. I see that the University of Calgary team has joined us, so I hope all is well with you guys. We have one more team that will be presenting, and then you guys will go after them. So I will pass it on to McMaster 3 to start when you're ready.
All right. So over to you, Fraz.
Thank you. Hi, my name is Fraz Chowdhury, and I'm joined with my friend Hamza Naqvi. Before we begin, we would like to thank the Del Maestro family and the selection committee for giving us the opportunity to present at this competition. Our proposal is titled Exploiting Communication-Based Pathways in H3 G34 DHT to Derived Receptor-Based Therapies. So H3 G34 mutant diffuse hemispheric glioma is a highly aggressive and infiltrative high-grade glioma, or HGG, that primarily affects a younger population. It is defined by a specific histone 3 and G34R/V mutation. It carries a poor prognosis with a 5-year survival rate of less than 15%. The standard treatment, which currently combines the canonical triad of surgery, radiation, and chemotherapy, has shown limited efficacy as these tumors are have significant resistance and are highly prone to recurrence. specifically genes such as PDGFRA, which is a cell surface tyrosine kinase receptor, has shown to be implicated in recurrence through a receptor-mediated pathway. One of the key limitations with current therapies is that they're centralized only to the core malignant populations of the tumor. However, the tumor is part of a much larger network and is surrounded by a rich variety of non-malignant cell populations. This includes, for example, the macrophages, astrocytes, or normal neurons. And each of these cells are involved in their designated communication networks with the core tumor population. And a few of these signaling pathways are, in fact, tumor progressive. So they help to drive the tumor. One of these examples of these pathways is the PDGFRA signaling axis. So as you may recall, PDGFRA is a cell surface receptor. So the cells in the microenvironment can exploit this cell surface receptor to drive tumor progression. This overall network is called the tumor microenvironment, or the TME, and it's part of a much larger concept known as intra-patient heterogeneity, which is within patient heterogeneity. So to design an effective therapy, we believe that the heterogeneous landscape of the tumor must be considered. And as such, novel therapies should ideally target both the core malignant tumor population as well as the tumor cells' communication with the surrounding microenvironment.
So how do we exactly plan on countering this issue of heterogeneity? We propose using single-cell RNA sequencing. So single-cell RNA sequencing works by providing an unbiased full transcript view of the gene expression data for each individual cell involved in our sample. And this provides single-cell resolution, not only revealing the populations involved, but also each individual cell's contribution. Think of it like the people within a city. Each person within the city contributes to the overall progression. So here we'll take patient samples collected from surgical resections and perform single-cell RNA sequencing. Then during our analyses, we'll cluster the tumor microenvironment cells based on similar features using bioinformatic tools, and then analyze the unique contributions of each cell, which we presume to be the ligand receptor interactions, and then develop antagonistic therapies, first using CRISPR knockouts and receptor antagonists in vitro and in vivo, and then moving it forward to clinical trials. So again, we're thinking of it as sort of a city. So here we have the HQ, the tumor cells, where we'll collect our samples from surgical resections from patients such as Jacob, as well as other adolescents suffering from diffuse hemispheric gliomas. We'll then use single-cell RNA sequencing to investigate the heterogeneity. Here we'll see the other buildings involved, and then be able to find out each individual working or living in these buildings. We'll be able to identify the names of these buildings and what their purposes are using bioinformatic tools such as SERRAT, which helps us to find the bank of immune, as in our immune cells, our endothelial hotel, which are endothelial cells, neuron heights, astrodome, etc., as well as our HQ, the tumor cells. And then we'll be able to look into the receptor ligand and signals, essentially who's talking to whom, using analyses such as cell-to-cell interaction analysis. We like to think of it as like a truck carrying a payload from one location into the next, essentially carrying the information needed for our end destination. So we'll end up taking the hits from our cell-to-cell interaction analysis and filtering out for hits accordingly. So say one of our hits was the PDGF to PDGFRA interaction. We'll validate this interaction and see if it has any sort of functional implications with tumor progression using CRISPR knockouts on in vitro models, with the in vitro model of choice being a tumor organoid, which has been the literature-backed practice for sort of modeling cell co-cultures. And we'll determine whether this disruption in communication causes tumor suppression.
We will then take the receptor-based targets that have been validated in AIM-2 and then further validate them in vivo murine models. And we will then take our top 10 promising candidates, which are safe and effective, to undergo synergic clinical trials. Ultimately, we expect that this will provide us with a more broad panel of therapeutic targets that attack not just the core malignant population of the tumor, but also work to impede the cell-to-cell communication that helps drive the tumor. We will then take this new panel and match this to the patients. In the matching phase, we will require single cell analysis to be done on patient biopsies to understand which communication pathways are upregulated or enriched for that specific population. Once again, considering the heterogeneity of the tumor, and then match that patient to a specific drug that we found in our synergy clinical trials. In terms of the overall timeline of the project, we expect this to take at least 10 years for the development phase. In this phase, we will map the tumor communication networks and validate our hits, once again, beginning with single-cell sequencing and analyzing those cell-to-cell interactions. We will then perform CRISPR-Cas9 guided knockouts, as well as receptor antagonism to understand if this is effective in an in vitro model, then validate that further in vivo models, and finally take this into clinical trial, which we think will take approximately 7 years or more. The application phase will be reduced due to the expansive development phase, where we will perform single-cell sequencing on patient biopsies, and within a month, we can get those results back from single-cell sequencing, understand those communication networks, and match those patients to specific drugs that we have found in our development phase.
So, in conclusion, currently therapies are limited by the tumor heterogeneity. So our solution is to essentially map these communication networks at a single cell level, going back to our model of the city. And then hopefully we'll be able to derive various receptor-based therapies that block tumor communication at its forefront. And by turning the tumor's own conversation networks against it, we can give patients more time and better outcomes. We'd like to thank the Maestro family and the Brain Tumor Foundation of Canada for allowing us this opportunity to present, and we're ready for feedback and questions.
Thank you very much. That was great. I'll pass it on to the judges.
Hi, it's Adriana here. Hi, Hamza Fras. Thank you for your letter describing your project and then this really great presentation. So as I understand, you're going to use single cell sequencing to look at tumor cells and also their nearby cells to see who's talking to whom, kind of identify the key communication signals, like the messages that the tumor depends on, and then try to block the chatter between them, and then find some promising targets for those communication signals. I think that's pretty cool. Like the way you broke it down, I think I understand your project better. And this kind of RNA sequencing is powerful, but I think it's resource intensive. Like we don't really use it routinely diagnostically. So how would you envision this technology being integrated? Like I'm looking at your timeline slide, into real world clinical timelines for children with aggressive gliomas like this, where we have to make decisions quickly. How would this I guess it's sort of a feasibility and clinical translation kind of question. Yeah.
That's a great question. Thank you for asking. You're right that single cell sequencing is normally done on a very expansive timeline. The analysis usually takes a long time as well. We expect that single cell sequencing will improve in the future. We know even right now that in setups where multiple samples are pulled together, the cost is reduced. The timeline is also starting to become smaller and smaller with more and more tools that we're using for the sequencing. For example, NovaSeq, which is often used for in vet lab projects to understand these heterogeneous models. So you're right that it is currently on a longer timeline, but with more technology, we expect it to be occurring on a smaller timeline. And with pooling of those samples, we expect that the cost will also reduce. But in terms of your question about its efficacy as a diagnostic tool, we want this to be sort of supplementing the initial diagnosis of the patients. So if the diagnosis comes back or the pathology comes back as a high-grade glioma of a specific type, and you want more details about which communication networks, for instance, are upregulated, then single-cell sequencing can provide that supplemental information to better help the neurosurgeons or the clinical team understand what is happening. So it is more of a on-demand diagnostic tool where if the timeline allows for further diagnosis and a timeline is able to integrate that into the clinical therapy as well, then we expect this to be a valid tool. But you're right in talking about the limitations of its timeline and also the cost as well.
And also, we've found that companies are already coming out with like faster and more like efficient and cost-effective ways, such as a company called Parse Biosciences. It essentially has combined and has improved upon previous like very costly procedures, such as the ones from companies like 10x Genomics, where it's created like a plate-based platform that uses more like cheaper like barcodes to this process called single cell combinatorial indexing. So that way you move away from these expensive microfluidics devices that like traditional methods have used, while being able to incorporate at a very ultra high throughput manner millions of different cells, meaning you can combine various different samples. And they have shared that their pipeline from results from sequencing all the way down to analysis can be done within a month to a month and a half because they've developed streamlined pipelines and streamlined workflows with this exact purpose of mind of diagnosis.
Okay, no, thanks for your answer, guys. That's great. It's very, very interesting, exciting prospect.
Any
Hey
other
guys,
questions?
yeah,
Yeah.
I have a couple. No, great presentation. I really liked your analogy with the sort of the buildings in the city of how to describe, single cell analysis and stuff like that. So, one question I had was, you're mentioning the tumor environment, microenvironment, and that is a very important consideration now with our treatments because it's not just the tumor cells communicating with each other, but it's also the immune cells, the neurons and astrocytes and all that that may be contributing to why these tumors are spreading and also developing in the first place. So have you guys considered a spatial transcriptomic approach rather than a full-on single cell analysis? Because looking at your analogy of the city, sure you can see how different cell populations and different groups communicate with each other, but in my mind, at least, single cell analysis by itself just doesn't have that ability to tell you where exactly things are happening, whether or not they're actually, you're at the brain tumor brain interface, whether that's communicating or whether there's any interactions going there. So I wonder if you can comment on that.
Yeah, for sure. We would like really appreciate the incorporation of a spatial with single cell. And we've seen that being applied in the glioblastoma region of brain tumors. So we would love to like also apply that here. It was just more of a resources sort of consideration since spatial transcriptomics at the moment is a little more cost intensive, but with further advances into that, it would obviously be a very like streamlined entry into our sort of pipeline where we already have these surgical resections, and then applying spatial transcriptomics to them would obviously further inform our analyses and perhaps streamline our later functional validations too.
Yeah, and just to add on to Hamza, just very briefly, this approach can be made more multi-model by incorporating different techniques. I think someone else talked about ChIP-seq before. There's a TAC-seq that also looks at chromatin accessibility. And then with spatial transcriptomics, we can sort of combine all these together to better understand those communication networks. So there is capacity to compound this approach with more and more techniques to better understand that network. But I think the underlying consideration is just how resource intensive that is and how fast we can get that diagnostic results.
Can I just ask one question, Sue?
Of course.
Maybe you could go back. Well, it's a comment in the question. Maybe you could go back to your AIM-1 slide.
Sorry,
No,
I
you've
just
already switched.
we just have University Calgary's presentation already keyed up for the next presentation, Joe.
Okay.
Do you want me to get them to unshare and reshare?
It'll only take a minute,
Okay.
and we've
All right.
got.
Is that the one with all the buildings, Joe?
Yeah, I have to make a comment on that because I'm like Ryan. I do kind of like the analogy with the buildings. Like when I saw that slide, to me, the endothelial hotel kind of looks like the Citigroup building in New York City, which I thought was neat. The Astrodome, of course, is not in New York City. I don't even think it exists anymore because it was in Houston, if I remember correctly.
Yeah.
Huron
that
Heights sounds like Huron Heights, which is here in London. The Bank of Immune Cells could probably be anywhere in the world. But I did think the Tumour Tower had a, kind of a striking resemblance to another tower in New York City. I believe it's called Trump Tower, and maybe you purposely picked that for reasons. But no, my just question is a simple one. It relates to the bullet point 3 there. Identify all tumor and surrounding cells using bioinformatic tools. Can you just explain that just briefly? Not
Sure.
too long. Yeah.
Sure. So essentially, when we receive the sequencing data, we get a sort of matrix of gene expression, gene counts, as well as levels of expression throughout, right? We can use bioinformatic tools such as SERAT, which have been streamlined in a way that you can go from start to finish, from quality control, removing out your sort of like low quality cells and multiplets, and then moving forward down the pipeline in terms of dimensionality reductions and clustering, to then finally move forward towards annotations, which we can do using various analyses such as differentially expressed gene analysis and gene set enrichment analyses. We can use that and combine it with automated annotation tools such as single R, which can be then used to sort of compound and like come up with our annotations. And then from there, we can use our annotated cell populations along with our gene expression data and ligand receptor databases to utilize our information and find out how the communications are essentially occurring. That's sort of like the sort of analysis pipeline that we would follow. And that's sort of a general summary
Great.
of
Thank
it.
you. That's helpful. Thanks. Thanks, Sue.
No problem.
All right. I'll
Excellent.
stop my sharing so then Calgary can get started.
Thank you so much.
Thank you.
Thank you.
University of Calgary, I just wanted to share that as it was a little bit challenging for people to be able to hear the alarm go off once your time was up, the solution was to just show a white piece of paper when you have 1 minute that's remaining. And then I will let the team know if your time has expired and you haven't completed that it's done so. And I'll do the same thing for the judges. Show the white piece of paper again when they have 1 minute that's remaining in the question period. So I hope all is well with all of you. And without further ado, I'll let you get started whenever you're set.
Sounds good. Thank you so much. And again, we want to apologize profusely for presenting late. We're incredibly sorry for that. Okay, Omar, take it away.
All right. Good afternoon, everyone. We hope you're all doing well. Now, what we are working here with today is a story of a young boy who has been diagnosed with a very aggressive brain tumor. His treatment, it's complicated. The options are many, but the answers, they're uncertain. But what my team and I have done today is bring you with a machine learning tool that transforms ideas into actions. It takes real data, learns from past children, and delivers guidance that can actually be used. Because at the end of the day, what matters most in research is that we have real solutions that are tangible, practical, and capable of sparking real change for children like Jacob. Now, of course, the first question is, how does machine learning relate to this? Well, on one side, we have the tumor. This is an aggressive childhood brain tumor caused by a mutation called H3G34. And what makes it difficult to manage is that its symptoms differ and the tumors present themselves in various ways. And so our aim is finding the right therapy for each child. It's like these glasses right here. Each person can have vision problems, but not each one will need the same prescription. And so what we are trying to do is provide each child their prescription, the treatment that gives them the best chance at health and clarity. On the other side, we have machine learning. It's like having a coach that studied thousands of past games. Instead of just looking at one move, it looks at the entire board, the tumor genetics, the scans, the treatments tried before, and so on. And from that, it predicts the best move for this child. In the middle, both sides meet, and we get a solution in a field where we see far too many families left with uncertainty.
Now, no tumors are the exact same. They are analogous to a fingerprint. But unlike a fingerprint, these tumors are spontaneous and malignant. Treatment must be tailored personally for each individual depending on the situation and the images presented. Even with advances in technology, with all the scans, hours of grueling work from world-class physicians, they'll face a problem of determining the correct diagnosis. How can this individual be helped, and will I be making the correct decision by applying this treatment? To that, I would like to bring you back to the year 1997, when IBM's Deep Blue shocked the world by beating chess champion Garry Kasparov. At the time, Garry was endorsed as being the greatest chess player in the entire world, yet this computer was able to outmaneuver him. The focus here isn't on chess, but it's on the computer's ability to hyper-focus on one specific niche, something us humans are incapable of doing. And just to reiterate, this was 1997. Fast forward to today, and computers have gone leagues beyond chess. And so with that, we are able to target whatever we're planning on targeting. Humans, no matter how skilled, are subject to fatigue, bias, and countless outside influences. And so, although computers aren't flawless, we're able to train them only on one specific thing, which in this case are glioblastoma genetics and imaging. And it's not swayed by the distractions that us humans are. It can stay locked in on this one task, turning complex data and the middle circle uncertainty into clear, reliable guidance for everyone.
And so, as always, we're left with a problem and a hypothesis. The problem is that just like we've been emphasizing, there is no clear way to match the right treatment to the right child. And the hypothesis going into this for us was that if we were able to use data from previous scans and tests, we would be able to find the best treatment for each child, including Jacob.
Now, here's Jacob's situation in plain terms. He's a 12-year-old boy and he has a fast-growing brain tumor in one of his hemispheres. Tests show an H3G34 change, and so this is essentially just a tiny switch in the tumor's DNA that pushes growth. And although surgeons have removed part of it, the tumor is able to thread into in between healthy cells, and so some of it is still there. And so the family and the team have to choose. Do we try another surgery? Do we go with radiation? Do we go with chemotherapy? Do we look for a trial? Each option has pros and cons. More benefit for some, while some side effects for the others. And so this is where our tool comes in. We feed it what we know about Jacob, what the physicians have seen, the MRI pictures, the H3G34 result, and how much of the tumor was removed, and we compare it to many past cases, right? There's thousands of cases being trained on this model. It then predicts which option is most likely to help Jacob and shows the reasons in plain language. So not only the doctor can understand, but his parents and caregivers and other individuals involved can make a confident, informed choice, ensuring that there is a less likelihood of error.
Now, here's how the model works. In an ideal setting, we would start by collecting and de-identifying multimodal data. Things like MRI scans, clinical details, genetic information, such as the H3G34 mutation, treatments received, and patient outcomes. Since we didn't have direct access to real hospital data, we worked with the publicly available dataset from Kaggle. This dataset included many similar types of information, including patient demographics, tumor characteristics, MRI findings, and survival outcomes. From the MRI-related information, we extracted features that represent patterns in the data, similar to radiomic features you would get from actual scans, which can capture details that aren't always visible to the human eye. We also mapped our treatments and outcomes so the model could learn how to connect. All this information was then combined to train and validate our model. The goal is for the model not only to predict the presence of a brain tumor, but in future, more complete version, help predict the best treatment option for a patient like Jacob, and importantly, explain which factors are driving those recommendations. Now, let's break down how the model was actually built. Think of the patient data as the ingredients in a recipe. Things like demographics, tumor size, growth rate, MRI symptoms, and prior radiation exposure. Before cooking, you need to prep the ingredients, so we standardize the numeric features, map the ordinal ones like symptom severity, and turn categorical data like tumor location or MRI findings into embeddings. These embeddings are like grouping similar flavors together, so the model can recognize which ingredients work alike. Once everything was spread, we trained a multi-output neural network, like a master chef with two arms. The shared layers are the chef's brain, learning patterns from all the ingredients, and then the two arms branch off. One predicts survival, like estimating how long a dish will take to cook, and the other chooses the best treatment, like selecting the right recipe for the ingredients you have.
Okay, so now let's talk about how the model actually performed. What we did was we had 50 epochs of training with mini batches of 32 patients, and the network's loss quickly stabilized at around 1.45. What this plateau actually tells us in practice is that the model reached a stable baseline and was consistently learning patterns from the data. Now, the survival prediction of the model achieved a mean absolute error of around 22.5%, which is incredible, and indicates it was capturing key trends and outcomes. On the treatment side, the accuracy was still great, but since that was just preliminary, we still see some errors like the model defaulting to chemotherapy, which reflects a limitation not in the model architecture per se, but rather just in the relatively small and imbalanced data set. What's most important here, and what we really want to stress, is that this system is a fully generated pipeline, including a UI, as you can see here on the screen, meaning that we've built a model and created a UI all within the span of just a couple of weeks. This shows that everything works end to end and shows that this could have utility in a clinical setting in the future if we were to make a more robust data set or collect our own, like we had initially proposed in our proposal. So altogether, even though the error can still be improved before it's actually be put to use, we still see a lot of benefit here. And this is just a couple of the pages that we wanted to highlight, with the main one being the prediction system. On the left-hand side, you can see that we have all the demographic data. And in the future, we want to use an image classification system, just so that way we can feed in images like what was given in with Jacob's case. And on the right-hand side, you can see the results with a predicted survival rate as well as a recommended treatment. Now, to conclude, I just want to reemphasize what my colleague said. Childhood brain tumors are some of the most aggressive and complex cancers we face. And because no tumors are exact same, personalization isn't optional, but rather essential. Thank you.
Thank you very much. I will open the floor up to the judges.
Well, I mean, I find this fascinating. I'm not quite sure I'm convinced of its usability. I mean, if you just go back to your last slide there, I think it's brain tumor prediction system. Yeah, the one before that, I guess. Yes, that one. I mean, it's interesting that the system, I guess, I don't know what to call it, BTPS, predicts that surgery is better, but not by much. In fact, I would argue that all four recommended treatment probabilities are about the same there. So essentially, a human has to then make the best guess of those four. Now, maybe that's because of the amount of data. I don't know. The other thing that I find difficult to conceive is how all the data that you need can be collected. Working at a hospital where data is in all different forms, imaging data is in one system, patient demographic data is in another system. Systems are constantly being upgraded and up. I mean, how would your machine collect all this data and be able to analyze it? Is that something easy to do for these machines, or do humans have to help with the data collection? Maybe it's just my misunderstanding of how you do these things. Take a shot at any of those questions.
Yeah, no, of course. I mean, you bring up a lot of interesting points. Like, The thing is, with a lot of AI models, especially predictive ones like this, I mean, obviously, none of these are yet at a point where we can use them in a clinical setting, and that's like an important distinction that obviously we want to make. And you're right, like for this, it's so close that essentially a human would have to still make the decision. But I think some of this is because of the fact that we didn't train on a lot of epochs, and we also, like I said, used like a very small data set. Obviously, the more that we train it, the more accurate it's going to be naturally. In terms of addressing the other part of your question regarding like different types of data, generally what happens with AI models is like all of it is converted to tensors. So essentially, regardless of the data that we're getting, we're streamlining it into one type. So there is, and there's always pre-processing, like Rayansh had spoke about. Even for the data that we got, pre-processing was done. So really, all we would need to do is change that aspect of the architecture, and the rest of it could remain largely unchanged.
That's great. Thanks. That's helpful.
I
If you
just
don't mind,
wanted
I'd like
to
to
say
hop
one
in
more
there.
thing. For
It's
like,
fine.
I know you talked a lot about like how we can actually accumulate all the data and then collect and then train the models on it. So what we initially planned to use in our letter of intent was the children's brain tumor network, which they have collected all of the data that we essentially needed for this project. But unfortunately, we had to like go through a specific process that they wanted to access the data since it's not really publicly available. It has to be done through like a proper university or faculty affiliated program. So that's why we weren't able to access that data, but it is out there. And eventually, like if we ever get the chance in future, we would like to train our model onto that data, which is way bigger and it has like all other specific things that we would require for the training as well.
I'd
Thank
like
you.
to very quickly add on just to answer that one question that you had. Yeah, humans would actually have to collect this data, like all the images and everything. It's not the computer collecting the data. It's the humans collecting that data and giving it to the computer itself. So we need to have like all the scans, all the demographic information put together, and then like the computer sort of gets the holistic overview of everything. Because right when you think of like a human being, right, you have like a stream of consciousness, but you can only think of like one thing at a time. So um essentially the computer is able to just take all those ideas, think of everything all together and put things together. And that's why we believe that this is the superior model.
Brian or Adriana?
Hi, it's Adriana here. Thank you guys for joining the research platform today. Even though you came later, I hope everything is okay. And I appreciated hearing your project as you've described it. I really appreciated your comparison to Deep Blue and Gary Kasparov. Do you know about that match? Do any of you guys play chess?
Yeah, we all play chess.
Oh, okay. So do you have a favorite chess opening? Do you know the chess opening that Deep Blue used to win against Gary?
Rayansh probably knows that.
Yeah, it's not relevant to the it's not relevant to your presentation. I was just curious. Or you could tell me later if you want.
Yeah, of course.
But yeah, I noticed in your letter of intent that you proposed building your data set from like public archives and published data. Um, I guess what I was really wondering about was, kind of the clinical integration aspect of this. So if your model predicted a therapy is optimal, but a multidisciplinary tumor board, I guess this kind of goes to Dr. Megacy. If a tumor board recommended another, how would you envision your tool being used? Like as a decision support adjunct or maybe more as a potential driver of the clinical choices for a patient?
I think one part of our model as well, sort of we tried to emphasize it in some parts of the presentation, sort of there's also an explanation part of it, right? So not only does it provide you with sort of, what it believes the best probabilities are, but it also explains why it actually gave those probabilities. And so in a case where, maybe the model gives one thing and, a tumor multidisciplinary board gives another thing, I think the model is still beneficial because it explains why it arrived to the conclusion it did, and that can be used in support to what the board actually, came to on their own.
So in a supportive role.
Yeah, in cases where it kind of conflicts, I think it has large, potential for supportive role because it explains why it came to the conclusions that it provided.
Okay.
Ryan, do you have any questions for the team?
Actually, that was similar to what I was going to ask
Okay,
anyways, but
perfect.
so I don't have any other further questions, but yeah, great job, guys.
Amazing. Thank you. Thank you so much to all the teams that took the time to submit your letters of intent, to prepare your presentations today, and to prepare accordingly. I'm now going to open up the breakout room for the judges to move forward to deliberate on making selection of the winners from today's competition. Also would like to welcome Dr. Westhaver, who joins us today to present while the judges are deliberating. I see that Dr. Roy is here as well, so welcome to both of you, and thank you for taking the time out of your day to present. Dr. Westhaver is a postdoctoral fellow at the Department of Surgery at Dalhousie University under the supervision of Dr. Adrian Weeks and Dr. Jeremy Roy. She completed her PhD at Dalhousie, and her research is primarily focused on differentiating patient outcomes in glioblastoma using immunological profiling. Dr. Westhaver is from Halifax, Nova Scotia, and is our 2025-2026 Done with Cancer Fellowship Award recipient. So welcome, Dr. Westhaver. And I see you've got your slides all set. So I will send the judges off into the breakout room and let you move forward whenever you're set.
Okay, thank you for the opportunity to present. I so appreciate it. I was asked to make this presentation accessible to a wider audience, so I'm hoping that it will be. And yeah, I'll get started. Oops, if I can present, okay. So glioblastoma or high-grade glioma is an aggressive form of brain cancer with an average survival of only 14 to 16 months, despite aggressive treatment, including surgical resection, radiation, and chemotherapy. After treatment for glioblastoma, patients have regular MRI scans to check for tumor growth. However, some patients can have MRI results that look like tumor growth, but is actually the treatment and the immune system reacting with the tumor. This is known as pseudoprogression. The problem is that pseudoprogression looks almost identical to real tumor growth on MRI. Because of this, doctors also often have to wait and repeat scans over several months before knowing for sure if the tumor is growing or not. So right now, there's no single scan or test that can clearly tell the difference. So we call pseudoprogression based on retrospective analysis. This waiting creates uncertainty, can delay important treatment decisions, and adds extra stress for patients and their families. What we do know is that the immune system, the body's natural defense against disease, may behave differently in pseudoprogression compared to true tumor growth. If we can better understand how the immune system responds to treatment, we may be able to use this information to improve care. Our research is exploring whether changes in the immune system measured through blood samples during treatment could help us tell pseudoprogression apart from true progression more quickly and more accurately. So researchers have begun to study how the immune system behaves in glioblastoma, and when we look directly at the tumor tissue itself, we see clear differences between true tumor growth and pseudoprogression. In fact, pseudoprogression often shows stronger signs of the immune system being active. The challenge, though, is that getting tumor tissue requires a biopsy, which is a highly invasive procedure. So that's why we're interested in finding ways to study the immune system through blood samples instead. So this type of what we call liquid biopsy is much less invasive and could give us valuable information about whether a patient is experiencing true tumor growth or just pseudoprogression. So accordingly, in our lab, we're collecting both blood and tumor samples from people with glioblastoma. This work has created the largest ongoing glioblastoma patient study in Atlantic Canada. So far, we have about 85 people enrolled as of fall 2025 and more than 200 blood samples collected. Blood is kind of taken at different points during the clinical journey. For example, before surgery, at the midpoint of surgery, shortly after surgery, and then about every 3 months when the patient returns for their regular MRI scans. At the time of surgery, we also collect tumor tissue. So together, these samples will help us study how glioblastoma and the immune system interact over time. So just to recap, essentially at every blood draw for each patient, we're collecting and freezing or cryopreserving the white blood cells of the immune system. This will allow us to study the immune system of these patients at any given time point during their clinical journey. So basically, we hope we can tell the difference between pseudoprogression and true tumor growth by studying the immune system. To do this, we look at different parts of the immune response. This includes immune cells in the blood, immune cells found within the tumor itself, and small messenger proteins in the blood called chemokines and cytokines that actually help the immune system communicate. So basically here I'm showing you a diagram of the white blood cells in the blood, the immune system. And to study the immune system in patients with glioblastoma, we use a tool called flow cytometry. And that lets us look at many different types of immune cells at once. We can also look not only at which cells are present, but how they're behaving. And we do this by looking at markers on the surface of the cells, which I'll be showing you in the tables on the right. So we have 2 panels of markers, each have 28 markers in them to study the immune system in the blood. In addition to the blood, we'll also be studying tumor samples from surgery to look at the immune cells that have infiltrated into the tumor itself. So just to recap, in our lab, we're building a growing collection of blood and tumor samples from people with glioblastoma. The blood samples are carefully stored at really, really cold temperatures. We call this cryopreservation, so they can be studied over time. And tumor tissue from surgery is used to look at the immune cells that are actually inside the tumor. We then stain the cells with fluorescent dyes that tag these markers that I told you about. And then we use a machine called a flow cytometer to read the signals on the cells. So flow cytometry allows us to study many different types of immune cells or white blood cells all at once. And today I'll be showing you some of the results from these analyses and what they can tell us about how the immune system responds to glioblastoma. So again, the tool we use to study the immune system in our lab is flow cytometry. And what I'm showing you here is just the data that comes off the flow cytometer machine and what it looks like. So each little dot you see here is an individual immune cell or white blood cell. And then we analyze the data to go from looking kind of at all the cells together to looking at specific populations of white blood cells using those markers that tell us who each cell is. So with this type of analysis, we can look at many different features on each cell at once. And we're looking at millions of cells at a time. So you can imagine this is a lot of data to look at all at once. And one way to look at all of the data all at once is to use a method called dimensionality reduction and clustering. So to give you an idea of how this analysis pipeline works, we start out with those dots that I showed you on the previous slide, where each dot is a white blood cell. We start by cleaning some of the data. This means removing anything that shouldn't be there, like dead cells or background noise, so that we're only looking at healthy, useful cells. Next, we add labels to the samples, almost like putting name tags on them. This way, when we combine everything together, we know where each piece of data came from. Then we merge the samples together. This is called concatenation. Then we use computer tools to simplify the data and make it easier to see patterns. So basically this kind of complicated graph with islands is looking at all 28 of those markers that I told you about all at once. We call this dimensionality reduction, taking 28 things or markers and putting them on a single graph. So after that, special programs group the cells into clusters based on their shared features, almost like sorting puzzle pieces into piles of similar shapes and colors. And finally, we study these clusters, the colorful kind of blobs on the screen, to show how the groups of cells differ across patients or conditions. So basically, all of this helps us to understand what's unique in each case and ultimately how the immune system might be behaving. So here I'm showing you one of those dimensionality reduction algorithms where you take all of those 28 markers or tags on the immune cell surface, you put them all into one graph to look at once. And here I'm showing you 5 different samples, so 3 healthy donors, 1 patient with a GBM, and 1 patient with a lymphoma. So you can see right away that using dimensionality reduction, it makes it easier to identify the differences between the donors. So basically, all I really want you to take away from this is that we can take this really complex data and see right away what the differences are between the patients. And what we can also do is look at each of those markers or tags that we put on the cells individually to see how the immune cells are laid out for each patient sample. And we can look at them using what's called a heat map, where the yellow and orange islands are expressing more of a given marker, while the green islands are expressing less. So in this way, we can look at really complex data and identify each population of white blood cells. And this is important because it's basically giving us a snapshot of what a patient's immune system looks like. And then we can compare that between people with pseudoprogression and true cancer progression. So here I'm just showing you those clusters again, now with their populations of white blood cells identified. This is for both of our panels of markers on the left and on the right. And basically all of the white blood cell populations we would expect in the blood are represented in our clusters. So again, this is giving us a snapshot of the entire immune system of what we call the peripheral blood, or the blood that circulates around the body. So basically it's a bird's eye view of how the immune system is behaving for any given patient at any given time point in their clinical journey. Okay, so now that we can see the whole immune system in the peripheral blood, we want to ask questions about how it's actually behaving. So again, using tools called dimensionality reduction and clustering, we get a snapshot of all the white blood cells in the blood. And now we can ask questions about how they're behaving in each patient and what they're actually doing at the time that the blood sample was collected. So if we focus just on the populations that are colored in, a type of white blood cell called T cells, which you might have heard about before in our earlier presentations, we can look at what they're up to in the blood. So in each of these little graphs, I'm just showing you one of those immune markers on the cells, and you can see that when we can compare the markers in healthy controls versus GBM versus that patient with a lymphoma. So again, all I really want you to take away from this is that not only can we see the whole immune system in the blood all at once, we can also get a readout of how the immune system was behaving at the time the sample was collected. Now I'm showing you a readout for a different immune cell population called gamma delta T cells. And here's a third population of cells called natural killer cells, what they're up to. These guys are actually really good at killing cancer cells in the body. So I won't show you all of the populations of all of the white blood cells for the sake of time, but you get the point. We're looking at all of the white blood cells in the blood and asking questions about how they're behaving, and also about how that behavior changes in patients with pseudoprogression versus patients with true progression of their cancer. Okay, so to recap, we've designed and optimized a tool called flow cytometry that allows us to get a profile of the entire circulating blood immune system in patients with glioblastoma. We're now ready to move forward and actually start analyzing our cryopreserved bank of samples that I told you about. What I'm not showing you today is that we've actually processed close to 60 of those samples in our frozen bank of cells. And what's exciting is that already we're seeing differences between the immune system in healthy people and patients with glioblastoma. And interestingly, also between patients that have pseudoprogression versus true cancer progression. So going forward, we'll be processing the white blood cells from the blood that we have banked, but we'll also be collecting and analyzing the white blood cells from inside the tumors themselves. So basically, when a patient has a resection surgery to remove tumor, we'll take the tumor and remove the white blood cells to study them as well. And then the third thing, as I said, that we'll be looking at is the messenger molecules that immune cells use to kind of talk to each other. So like text messages between immune cells. So we'll be testing the blood to ask what kind of messages the immune cells are actually sending each other. So in summary, we're studying the immune system, which is the body's natural defense against disease and cancer, and looking at how the immune system is different in healthy people versus individuals with glioblastoma, and specifically how the immune system differs in patients that have something called pseudoprogression versus patients with cancer that is actually progressing. So we take blood samples from individuals with glioblastoma and from healthy people to compare to, and we freeze those samples at really, really cold temperatures called cryopreservation. This way we can thaw the cells when we're ready to study how the immune system is behaving in all of the samples. What we'll also do, in addition to looking at the cells in the blood, is to take the tumors and extract the white blood cells from inside them and see how the immune system is behaving inside the tumor. We'll then tag all of those immune markers on the white blood cells and read the samples on that machine I told you about called a flow cytometer. We'll then use computer programs to analyze all of that complex data in a way that lets us visualize the whole immune system at once, like a snapshot of how that patient's immune system was behaving when the samples were collected. Finally, we can ask questions about how one person's immune system is different from another, like how they differ between healthy controls and patients with glioblastoma. We can also ask questions in real time with flow cytometry about how the immune system differs in patients who have pseudoprogression versus true cancer progression, with the hope that we can use the immune system to tell the difference between them. This will improve clinical decision-making and quality of life for individuals with glioblastoma. So I'd like to say massive thank you to Dr. Weeks and Dr. Roy. I've had a really, really great experience in their lab. They're absolutely lovely people and fantastic supervisors. Thank you also to everyone who makes the research of the Weeks Roy Lab possible. Thank you to our sources of funding. In particular, I'm funded by the Done with Cancer Brain Tumor Research Fellowship from the Brain Tumor Foundation of Canada, and Give to Live through the Cancer Research Training Program of the Beatrice Hunter Cancer Research Institute. And most importantly, thank you to the patient partners and their families, without whom none of this would be possible.
Thank you very much, Lauren, for taking the time to join us today and present on the important work that you are doing. It's a great inspiration, I think, for the students that join us today, seeing all the hard work you've put into getting to where you are at today. And we're excited to hear more about your project as it progresses and your continued involvement in our organization as your career progresses. So I'll open up the floor to anyone who is participating today, any of the students, if you have questions for Dr. Westhaver. And I'm not sure, Dr. Westhaver, if you're open to questions also about, kind of how you got to where you're at today, if the students are kind of deciding pathways.
Of course.
And to ask any questions that they might have in terms of their own exploration. So if there's any questions that the audience has, please feel free to unmute yourself and ask those questions now. Yeah, go ahead, Hamza.
Hi, great
Hi.
presentation.
Thank you.
I'm currently in a lab that works with like the onset of like co-cultures between gamma delta T cells and glioblastoma, and specifically patient-derived glioblastoma with like recurrent tissues.
Oh, neat.
So essentially, my question is like, what brought you guys to like flow cytometry and not like something like a sequencing method or like attack sequencing or like ChIP-seq, etc. Like, why flow instead of like those?
So, I mean, the answer really is that I did a lot of flow in my PhD and I know
Yeah,
how to do flow.
yeah,
But I think also flow is an incredibly powerful technique.
yeah,
So you're actually differentiating the cells so we can look at a single cell level using flow, and it's a lot less expensive.
yeah. So do you guys like fact sort it beforehand, or do you
Nope,
guys?
nope. So we take the peripheral blood, we run it on a fivefold gradient,
Okay,
basically, and then the cells get cryopreserved, so they get frozen, and then
okay,
they get thawed, tagged with antibodies that
yeah.
mark each immune marker on their surface, and
Mhm.
then you just run it on the flow cytometer, and you can resolve it kind of at a cellular level to see who's doing what and who's within the population. So
Oh,
we
neat.
have a pretty broad immune cell panel, so that we can look at all of the cells minus the granulocytes, which don't
Yeah,
really cryopreserve
yeah,
well, of course. So
yeah, yeah. Neat.
do you guys find it hard to isolate gamma delta T cells for such a low population?
And what we end up doing to like counter that is like essentially we expand them and activate them using cytokines like IL-2 and IL-15, and then
Gotcha.
expand it with another chemical in a way called zoledronate, which sort of like in accumulates phosphoantigens, and that way
Okay.
that uses like since gamma deltas recognize stress ligands like uh, IPP. I forgot the exact a full name of it, but through that we end up like inducing their expansion, and then we've been checking it based on either in non-transduced with non-transduced cells, as in just expanded and activated gamma delta T cells. With glioblastoma, we've seen that sort of there's a clear like suppression mechanism happening like within like 30 minutes to like 4 hours, where immediately we see a reduction of cell-to-cell interactions. These cells are already like starting to behave differently and all of that. So like with like transduction with like specific like CAR receptors and like all that, we see improved responses. But right now we're trying to figure out like how do we like properly improve it and like prolong the response of these gamma delta T cells, since they are more of an innate type of population.
Right. Yeah. It's neat that you're studying them. I don't think they get enough love as immune cell populations. They're kind of
Yeah,
the forgotten middle child of immunology. But
exactly. So my supervisor is quite interested in them because of their promise as like an off-the-shelf immunotherapy.
right, right. You don't have
So
to worry about histocompatibility, I imagine. So
exactly. So now we're just trying to investigate and see how we can best apply that to like the scenario of like glioblastoma and various other gliomas. So
very cool.
it goes, but I was really intrigued by your talk, and I will like probably like like nudge them a little, be like, hey, can we try this? Thank you though.
Thank you so much. Dr. Roy, go ahead.
Thank you. I'd just like to follow up on that question. So, included in Lauren's panel, and I think she definitely undersells this panel because it's actually quite extensive. I don't think anyone out there has done a 56 marker panel for GBM or for brain cancers. It's quite maybe CyTOF or other types, but this one's pretty good. So in her panel, she has uh, 9 different functional markers. So there's 6 markers of function, 3 markers for inhibition, including like PD-1, TIGIT, and TIM-3. So kind of touching on that question, she can then identify the function or how much, if these immune cells are functionally active, if they're exhausted, for example, if they're being inhibited, and that kind of thing. So I think one of the themes of her results is we're actually seeing exhausted cells, which is a hallmark of GBM, a whole immunosuppressive environment in the periphery for GBM patients, which again is like a hallmark of GBM. And so I guess going along the same line, so are these gamma delta T cells, what's their functional status? I think we could answer that question as well. So it's not only her panel doesn't just say there's this number of cells that are there or this number of cells is different. It's really about how the activation status or inactivation status of the immune system. So it's going to be really it's going to be fun to publish this.
Yeah, it seems like really interesting findings, and I'm sure the people in my lab would also be very interested because a lot of the others do a lot more like flow than I do since I'm an undergrad, so I haven't been given that sort of
Mhm.
exposure yet. But yeah, like I know that for example, the postdoc that I shadow, she would probably be quite interested in it because like she's also like someone that's quite interested in like using flow and like incorporating it into like uh glioblastoma as well and like these co-culture models with these T cells and engineered T cells because yeah we've
Very
seen
cool.
this situation of like exhaustion and like clear like suppression of like mechanisms but being able to characterize it at such a like vast panel is something that we'll be very curious to see like how to apply it, so very excited for that publication.
We are too. I see that the judges are back, and thank you for your question. I'm just going to drop my email in the chat, and if anyone has any additional questions, please email me, and I'd be happy to answer.
Amazing. Thank you again for being here and taking your time today. Thank you to Dr. Roy for being here. Again, we look forward to seeing your project continue to move forward and the results to come. So without further ado, we're excited to share the results of today's competition. And I'll ask that Dr. Wang shares the judges' selections, starting with third place, second, and then first place teams. So Dr. Wang?
All right. Well, thanks, Sue. Yeah, no, before I present our results, I'd like to thank everyone for being here today and for presenting their projects, and of course Sue and the Brain Tumor Foundation for putting this together. I've been doing this for a couple of years, and every year I'm impressed by the amount of effort that you guys put into these presentations, the creativity, and also just how well put together your presentations are as well. So, regardless of what the results are, congratulations to all of you for being selected and for presenting your projects. I think all of you have very bright futures if you choose to continue either, to medicine or neuro-oncology research, whatever you choose. So now I'll go to our results. So I'll start with third place, then go second, and then first. From what we selected for our third place recipients is Calgary 1. So that's Zehan, Omar, Reinash, and Meherab. So congratulations to you guys. You're third place recipients. Our second place recipient is McMaster Team 3, Hamza and Kras. Congratulations to you too. And then finally, our first place recipients is McMaster Team 2, Veer and Vanny. So congratulations to you both. And we're very well done.
Yeah, thank you so much.
Thank you so much, Dr. Wang, and congratulations to the winning teams and to everyone who took the time to participate today, who submitted through letters of intent, and to those who will move forward. We're excited to see your names cross our way in the future and other opportunities as we move ahead and as your educations and careers evolve. Thank you again to our judges, teams, the participants, the co-founders for making today's event possible and another great success. We're grateful to be able to offer these events and encourage you to explore other events and opportunities, volunteer opportunities and supports that we offer. We look forward to continued connection and supporting you throughout your future. Thanks to events like this competition, with determination, dedication, and commitment, we can work together to make a greater impact. One last reminder, I'd like to share with everyone that on October 24th is Brain Cancer Awareness Day. We invite you to raise awareness by joining our Hats for Hope campaign. We currently have toques, mittens, or hoodies for sale. I encourage you to share a photo of yourself on social media on October 24th wearing one of our hats or a hat of your choice using the hashtag Hats for Hope. I will be in touch with any of the winning teams as well as the other teams that presented today. And again, we thank you for your time, your dedication, and we hope you have a great remainder of the weekend. So without further ado, we will bring the meeting to close, and thank you all so much for all of your time and dedication.